Alright, now we're going to look at the regulation of glycolysis in liver. Again, we're going to look at PFK, hexokinase, and pyruvate kinase. These are the three steps that have a negative delta G in glycolysis. First, we have to look at the role of the liver. It has a more diverse function. The role of the liver, one of the roles of the liver, is to maintain blood glucose levels. The liver will store glucose as glycogen when glucose is plenty, and it will release glucose when the concentration is low. It also uses glucose, to generate reducing power for biosynthesis. So because the liver has these diverse roles, the regulation of glycolysis is more complex. So let's look at PFK1 regulation in the liver. So regulation with respect to ATP is the same. As what we saw in the muscle. If we have high ATP to AMP, we have inhibition, and if we have low ATP to AMP, we have activation. Glycolysis also furnishes carbon skeletons for biosynthesis, and so a signal indicating whether building blocks are abundant or scarce should also regulate phosphofructokinase in the liver. So PFK in the liver is going to be inhibited by citrate. And we will explore the structure of citrate in more detail when we look at the TCA cycle. A high level of citrate in the cytoplasm means that biosynthetic precursors are abundant. And this means that there is no need to degrade additional glucose for this purpose. So citrate will inhibit PFK by enhancing the inhibitory effect of ATP. One means by which glycolysis in the liver responds to changes in blood glucose is through the signal fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is an activator of PFK. In the liver, the concentration of fructose 6-phosphate rises when blood glucose concentration is high. And the abundance of fructose 6-phosphate accelerates the synthesis of fructose 2,6-bisphosphate. So hence, an abundance of fructose 6-phosphate leads, increased fructose 6-phosphate is going to lead to increased fructose 2,6-bisphosphate. The binding of fructose 2,6-bisphosphate increases the affinity of PFK for its substrate. So you can see that in the two graphs here. When we have PFK, and when we have fructose 2,6-bisphosphate, as we increase the concentration, you can see by this change in the curves that we increase the affinity for the substrate. So let me write that down. As we increase fructose 2,6-bisphosphate, we increase the affinity for fructose 6-phosphate. And you can see that as we go from a sigmoidal curve to a Michaelis-Menten curve to an even steeper Michaelis-Menten curve. What these curves are showing you is that it takes less substrate to reach Vmax, or maximal velocity of the enzyme. Right, if we look at the second graph, we can see that the binding of fructose 2,6-phosphate to PFK, it decreases the inhibitory effect of ATP. So you can see as we increase the amount of fructose 2,6-bisphosphate, we almost eliminate the effect of ATP. We need higher and higher concentrations of ATP to inhibit the enzyme. And this ties into the papers that we looked at, particularly the Cold Spring Harbor paper, which looked extensively at fructose 2,6-bisphosphate and its role in activating PFK. All right, just to summarize for everybody, so in the liver, PFK, okay, we are going to be inhibited by high ATP, low AMP. We are going to be inhibited by citrate. We are activated by low ATP, high AMP. And we are activated by fructose 2,6-bisphosphate. If you remember from those papers, we'll talk about them, and fructose 2,6-bisphosphate, but this was synthesized by PFK2, which had both a kinase and a phosphatase domain. All right, now let's look at hexokinase in the liver. All right, hexokinase in the liver is controlled as it is in the muscle. So it's inhibited by its product, glucose 6-phosphate. Now, the liver, in keeping with its role as a monitor of blood glucose, also has an isozyme of hexokinase called glucokinase. Now, this enzyme is not inhibited by glucose 6-phosphate. Glucokinase phosphorylates glucose only when glucose is abundant. Because the affinity of glucokinase for glucose is 50-fold lower than hexokinase. Glucokinase phosphorylates glucose so it makes G6P for glycogen and for fatty acid synthesis. Because it's low affinity, it allows the brain and muscle access to glucose when it's limited. It also prevents its waste when abundant, because you can make glycogen. The beta cells of the pancreas also have glucokinase. Now, if you remember, the pancreas secretes insulin. So, when glucokinase makes G6P, insulin is secreted, and the body will take up glucose because glucose is abundant. And this signals the formation of glycogen and fatty acids. Alright, finally we come to pyruvate kinase. Okay, and similar to the muscle, we have inhibition by ATP and alanine. What happens in the liver, though, is that we can also have inhibition based on covalent modification. And here we're looking at a phosphorylation reaction. And so in the liver, the enzyme can be controlled by reversible phosphorylation. And this is done under hormonal control, so that when glucose is low, glucagon is secreted and the enzyme is phosphorylated, which is going to decrease its activity. This prevents the liver from consuming glucose. When it's limited in supply. And so this is an example of how we can have different isozymes in the muscle and the liver based upon their biochemical role.